Hey guys, I just wanted to create a video to kind of clarify some information for you about this week's activities. So if you are on the Zoom call with us on Monday, we went over DNA replication, which is the process where one DNA molecule completely unwinds and creates a second completely separate piece of DNA. Now remember, when we did that, when we unwound DNA, we had one side form one new DNA molecule and the other side form the other. So you ended up with two new DNA molecules, but each had part of your original. So when you go through DNA replication, you're creating an entire DNA strand. You're making a complete copy. So the rest of this week's activities had to do with transcription and translation. And transcription, which is the first POGL that you were supposed to work on this week, has to do with creating what we call mRNA. And the reason we would make mRNA in the process of transcription is that DNA is trapped inside the nucleus. It's too big. So this is your nucleus of your cell. Your DNA molecule can't get out, but when we want to make a copy of some sort of instruction from DNA, what we do is we create this single-stranded mRNA molecule. The M stands for messenger RNA, and then that single-stranded molecule is able to leave the nucleus. So I'm going to kind of switch diagrams over here to give you this diagram, and in this diagram you can see that essentially we take this giant DNA molecule that's taking up my entire page, and what I've done is I've only opened up one small segment. So I'm going to only open up what we call one gene. So that could be, say, you want to eat a lot of ice cream, so you want to break down lactose, which is just a sugar. So you need to make a lot of copies of the enzyme lactase. So in your DNA strand are instructions somewhere in one of these little bends to make lactase. So what's going to happen in the process of transcription is we're going to open up, we're going to separate, helicase is going to come in and open up just this one little chunk, which say this is our lactase gene, and you're going to copy the message on one of these sides. Now, doesn't matter if it's the antisense or the sense, if it's the 5' or the 3', it doesn't matter because I'm going to get the same message on either side. So essentially what happens is we unzip this DNA, we break all these hydrogen bonds, you have your spare nucleotides that come in, and then they're going to find where they belong, and RNA polymerase, what it's going to do is it's going to hook these sugars and phosphates together on the side of your mRNA strand that you're building, and it's going to separate those bases from the DNA strand. So essentially what's going to happen is this single-stranded RNA is going to leave your cell. So I'm going to switch over to the POGL that you were working on this week, and you can kind of see this in a big picture form. So the questions I always get if we were in school, you would say to me things like, where in the world are these free nucleotides coming from? These are things that we get by eating different types of food. So your body breaks down the food you eat, and you have these things kind of floating around in your cell all over the place. Now, again, this is inside the nucleus as it points out here. So you can see big picture here. We're opening up just one little bump here on DNA, one gene. We're having these nucleotides come in. Now keep in mind we're making RNA this time. So we're back on this last diagram that I showed you, where there was an A on DNA because the outside is the DNA. Notice that A was originally bonded to T, but because we're making RNA, we've replaced it with U. So then the T bonds with A, C's go with G. So really the only switch between RNA and the DNA is that we're bonding a U here this time. So other than that, it seems really similar to what happened in DNA replication, but it is very different because we're only copying one side. So you see these molecules come in, you can see that this guy kind of is coming off here, and then it's going to leave your nucleus to go on. So essentially what we're doing is transcription. You're making the script. So imagine if you were transcribing something, you're writing down the instructions. So this is the transcript that then we're going to take outside of the nucleus for translation. So I'm going to go back to this original picture. So DNA replication and transcription both happen in the nucleus. Then what happens is your mRNA, your messenger RNA molecule, is going to leave your nucleus. What it's showing you here in this picture is this bubble thing here. This is actually a ribosome. And what's going to happen is that ribosome kind of has two parts, and it like clamps right on to your DNA or your RNA molecule. So it's actually going to, if you think of this ribosome as kind of like a sewing machine, and this mRNA molecule is kind of like the fabric that you're going to just feed it right through that ribosome. So basically what you're going to see happen now is that in the process of translation, we're going to take the code. So in this picture here, and I've kind of drawn some lines, this comes from your POGL that you have on page 3. This bottom thing here is our mRNA strand. So maybe this is our lactase instructions that we made from transcription. So then what's going to happen is our ribosomes that, as you know, is a part of the cell, are going to just kind of clamp onto this at the start codon. And the start codon is AUG, always the start codon. And basically what happens is your mRNA molecule, once you find a start codon, you're going to break it into sets of 3, which are called codons. And then what's going to happen is the ribosome knows, so it's going to attach on the start codon, and what that does is then it calls in these tRNA molecules. So this is a charged tRNA molecules. T stands for transfer RNA. And these are little amino acid taxis. So you know that the building blocks of protein are amino acids. And remember that we've talked about how proteins work with a lock and key fit. So each of these little tRNA molecules is going to have its own little code on the bottom. These are called anticodons, and the anticodon matches up with the letters in the codon. And each of these is calling for a specific little amino acid. So what's going to happen is we get AUG, and you could figure out which letters go with that. So A goes with U goes with A, G goes with C. So this tRNA molecule knows it belongs here. It's going to go to the ribosome. It's going to line up. Then what's going to happen is the next guy is going to come in. So then we know that we're now looking for C is going to be our next one. So that would be this guy over here. So basically this tRNA molecule would then come in and line up right there. So then what the ribosome is going to do is it's going to check that these codes match, and it's going to grab this little amino acid from it. So I'm going to switch pictures for a second. And you're seeing the ribosome is kind of building this little amino acid chain. And you're going to see like this was our first one, tRNA molecule. This was our second one. And notice that they no longer have their shape up here because they've given their amino acid to the ribosome and they're building this amino acid chain, which is going to be our protein eventually. So in the so the initiation is where it starts when that ribosome connects to that mRNA molecule at the start codon. Elongation is when we start to form this amino acid chain. And so basically tRNA molecules are kind of like amino acid taxis. They know that they are picking up a specific amino acid based on the code, their anticodon. So this keeps happening until we get to termination. So you can see all these guys have dropped off all of their amino acids. We've built this little chain, and what we get to is called a release factor, and that's called a stop codon. So all of the code for this is in our mRNA molecule that came from transcription. Now, on the POGL, the reason I sent you a PDF version is this codon chart can be mighty confusing. And in the original version that I posted on Schoology, this was actually kind of shifted, so everything was out of order. So the first thing I want to let you know is this is a codon chart. So you're looking for the codons which are found in mRNA, not tRNA. So essentially, what you're going to see is your first base, second base, third base is how you use this chart. So I'm going to kind of go back to that other sheet that we had going on here, and I'm going to show you how to use this. So our first codon was AUG. So to figure out what amino acid AUG would be, is I would go, my first letter is A, my second letter is U, and then I'm looking for the G on the side, so I know I'm in this second box here. So AU meets in this box, and then the G is on the very bottom. AUG gives me the amino acid MET, which is known as my start codon. So then the second code here was UUG. So to use the chart, I'm going to go U is here, so I know I'm in this box, and I'm looking for UUG, which is this bottom one, which gives me the amino acid LEU. So that's how you use this chart to figure out which of these amino acids is getting drawn in. So essentially, I can give you an mRNA code, and you can figure out exactly what the sequence of amino acids is going to be that got brought in. So for example, On the POGL on page 2, using the mRNA codon chart in Model 1, complete the following. So if I knew DNA was TAC, I know that if I'm making mRNA, I'm going to go A, U, G, G, A, G, C, C, U, A, C, C, A, G, U, G, A. Now, notice anytime I thought that I should maybe attach a T because I was making mRNA, I put a U in there, and then the C's and G's are good. So when we talk about the polypeptide sequence, this is where I'm going to take my mRNA molecule and I'm going to figure out this with the chart. So again, AUG was my first one, so I know that's MET. My second one is GAA. So GA, so I know I'm in this box. A is right here, so this is GLU. GCC, so GC, and then in the bottom box, C is ALA. And these have longer names, but guys, you don't want to know them. The next one is UAC, so UAC is TYR. CAG is CA. G is right here, so this is GLN. And then ACT, so, oh, not ACT, I'm looking at my mRNA, so UGA would be UGA, and look at that, it's a stop codon. So what that stop codon does is it says to the ribosome, hey, hold up, you're done, stop attaching things, and it's going to break off, and it's going to leave. That protein is going to then go on to do its job. So if it was lactase, it could go on and find the sugars to help break down. Say it was, you know, the protein that's used to build your muscles and a muscle fiber, so it would go do its job. So essentially this is important, and something that you should include in your video is the fact that, like, if I want to produce some sort of characteristic in my body, some sort of structure in my body, I'm going to have that code in my DNA molecule. And then if I need to get that code out of DNA, I do that through the process of transcription. So I can take the message of DNA, I can make it into a shorter message so it's easier to transport. I make a script through transcription in the nucleus. Then that script, mRNA, which is what I'm making in transcription, leaves the nucleus. It finds a ribosome to go through the process of translation. The cool thing is these mRNA molecules, once you make them, they can be used over and over and over again until something damages them, and then you would go back through the process of transcription to make another one. I hope this helps, and I hope you have a great weekend.